Hey everyone. In this video, I'll take you through a thought experiment and a real experiment that nicely illustrate the effect of sexual reproduction on the evolution of species. We will continue to address one of the questions from the last video. How does sexual reproduction drive adaptive evolution? Since adaptive evolution involves an increase in fitness, let's try to answer the question, how can sexual reproduction increase the fitness of a population or species? We'll do this by doing a thought experiment, which is essentially just imagining what would happen in a certain situation. A thought experiment is used to explore the potential consequences of a principle or phenomenon. In this case, the consequences of sexual reproduction, specifically crossing over, independent assortment, and random fertilization, on the fitness of populations, and here in the presence of two viruses. Thought experiments also often involve simplifying assumptions or idealized conditions. Here, we'll compare one asexual and two sexual populations, and assume that these populations are otherwise identical, and that no mutation takes place, and that each population has 4 individuals with the same 4 genotypes. Now let's say that the A gene has 2 alleles. A1 is resistant to virus 1 and is recessive, and A2 provides no resistance and is dominant. Likewise, the B gene has 2 alleles. B1 provides no resistance and is dominant, and B2 provides resistance to virus 2 and is recessive. Therefore, only the individuals with the A1A1, B2B2 genotype are resistant to both viruses. None of the individuals in our 3 populations are resistant to both viruses. When each individual in the asexual population reproduces, the offspring are identical. Now, let's say that both virus 1 and virus 2 enter this population. None of these genotypes is A1A1, B2B2, so all are susceptible to the virus and die. And the population goes extinct. Why did this happen? Well, in this thought experiment, no new genotypes were produced, because asexual reproduction usually produces individuals with the same genotype, and the existing genotypes were not resistant. Of course, we saw an exception to this in the last video, where asexual bacteria could quickly produce genetic diversity, but let's assume that these organisms are animals and not bacteria. Now, let's consider a sexual population where the A and B genes are on the same chromosome. Let's say that cells from these 2 individuals here enter meiosis, with the A1 and B1 alleles on the same chromosome, and the A2 and B2 alleles on the other chromosome during prophase 1. If crossing over occurs, it can produce recombinant chromatids with an A1 allele and a B2 allele. Each of these chromatids is sorted into gametes during the rest of meiosis. Recall that random fertilization says that any sperm can fertilize any egg. So, if this sperm and this egg happen to fuse, they will produce an offspring with the genotype A1A1, B2B2, which is resistant to both viruses. In the presence of these 2 viruses, this individual will survive and reproduce, and this genotype will become ever more common. Eventually, much of the population will become resistant to both viruses. This constitutes adaptive evolution because the population will be better suited to the presence of both of these viruses. Without crossing over, we would never have gotten this genotype because the A1 and B2 would have remained on different chromosomes and couldn't be inherited together to produce an A1, B2, B2 individual. In this example, the A and B genes were far apart on the same chromosome. But what if they were right next to each other? In other words, what if the genes were closely linked? In this case, crossing over between the A and B genes is unlikely, so therefore, no recombinant chromosomes would be produced, preventing the production of the A1, B2, B2 genotype. So resistance to both viruses at once would not arise in this population. So gene linkage reduces recombination between the genes and prevents some genotypes from being produced. Keep in mind that the production of new genotypes by crossing over is more or less random. The resistant genotype is not produced in response to the viruses. It occurs randomly. And if a genotype produced by crossing over just happens to be resistant, and if both viruses just happen to be present, only then is that genotype selected for. Now, there is some evidence to suggest that in some species, the rate of crossing over might increase in response to environmental stress, but which genotypes are produced is still probably more or less random. Now let's examine a sexual population where genes A and B are on different chromosomes. Again, let's say cells from both of these individuals undergo meiosis. Note that both these individuals happen to have the same genotype. Because the genes are on different chromosomes, and because we're only following two chromosome types here, there are two possible ways that chromosomes can line up during metaphase 1. As the cell proceeds through meiosis, different possibilities produce different gametes, depending on how the chromosomes line up. In other words, independent segregation of chromosomes makes the two possibilities equally likely so that different gametes can be produced. This occurs because the genes are on different chromosomes. Again, during random fertilization, perhaps this A1B2 gamete just happens to fuse with this A1B2 gamete, forming a recombinant A1, B2, offspring, which is resistant to viruses 1 and 2. The spread of this resistant genotype constitutes adaptive evolution. Like crossing over, independent assortment randomly produces new genotypes in offspring, so this resistant offspring was produced by chance, not because viruses 1 and 2 were present. In this simple example, we're only keeping track of two chromosome types, a large one and a small one. The number of chromosome combinations in diploid organisms is 2 raised to the n, where n is the number of chromosome types. If there are 2 chromosome types, 2 to the second equals 4 possible combinations of chromosomes in the gametes. And that's what we see in our gametes here. Here's one combination, a second, a third, and a fourth. But humans have 23 different chromosome types. That's 2 to the 23rd, or over 8 million possible chromosome combinations. And assuming no crossing over 8 million possible genotypes in the gametes. When you take into account the extra genetic diversity caused by crossing over and random fertilization, you have an almost infinite number of gamete genotypes that could be produced by sexual reproduction. Which is why no two people on Earth, except identical twins, have the same genotype. So, all other things being equal, sexual reproduction through crossing over, independent assortment, and random fertilization produces more random genetic diversity in the form of new genotypes. One of these genotypes, or more, can be selected for if those genotypes happen to be beneficial under the population's environmental conditions. Here, the presence of 2 viruses. Resistance to both viruses could still evolve in our hypothetical asexual species, but it would probably take longer than in their sexual counterparts because it requires rare beneficial mutations to occur. Now, I hope you found this thought experiment useful, but now let's turn our attention to an actual experiment. This experiment was both elegantly designed and carefully executed. Determining the impact of sexual reproduction on fitness is more difficult than you might imagine. One approach is to have an asexual and a sexual population that are otherwise similar and compare how fast fitness increases in the two populations after an environmental change. Another approach is to take 2 identical populations and alter one of them so that there will be no recombination in the form of crossing over and/or independent assortment, and allow the other population to undergo recombination, and then compare how fast fitness increases in these 2 populations. This is far easier said than done, and researchers have taken both of these approaches, but let's examine one study that took this second approach. This experiment is from a study from 2001 authored by Rice and Chippendale. Be aware that I'm going to greatly simplify what was done in this study, but I'll put a link to the original paper below the video if you're interested in reading it. The hypothesis was that sexual reproduction, particularly the independent assortment of chromosomes, can increase the fitness of a population. They tested this by producing fruit flies that either could or could not undergo recombination by independent assortment. They accomplished this by manipulating the chromosomes. They produced a synthetic X chromosome with a W+ allele on it. This allele confers red eyes. They also produced a synthetic Y chromosome with a W+ allele on it. The way that they did their crosses, the X chromosome will undergo recombination, but the synthetic Y chromosome will not. In this experiment, the authors cleverly took advantage of the fact that crossing over does not occur in male fruit flies, so the only recombination that was occurring was due to the independent assortment of chromosomes. This recombination from independent assortment only occurred for the artificial X chromosome. There was no recombination or independent assortment for the W+ alleles on the synthetic Y chromosome because it was fused to other chromosome types, making a giant chromosome, so that all the alleles of those chromosomes would segregate together, and wouldn't be able to make new combinations of alleles through independent assortment. While in the recombination flies, the chromosomes remain separate, so independent assortment could occur. In other words, offspring could inherit the X chromosome with the W plus allele at the same time as inheriting other chromosome types with other beneficial alleles on them. Flies with either the recombining chromosome or the non-recombining chromosome were introduced into 17 different populations of white-eyed flies, white eyes being conferred by the W minus allele. The W plus allele was selected for in these populations, and the change in the frequency of the W plus allele was measured over 11 generations. In both the 17 populations where W plus could recombine with other alleles, and in the populations where the W plus allele could not recombine with the other alleles. Since the W plus allele was the higher fitness allele, its frequency increased in both the recombining and non-recombining populations. But the recombining populations showed a faster increase in the frequency of the W plus allele. That was the main result of this study. Now, let's interpret it. Recombination, in the form of independent assortment in this experiment, created beneficial allele combinations in only the 17 recombining fly populations. This increased the speed of selection of the W plus allele, because the W plus allele could combine with other beneficial alleles, creating flies with even higher levels of fitness. Selection of these flies resulted in an even faster increase in fitness and a faster increase in the frequency of the W plus allele compared to the non-recombining populations. So recombination more quickly increases the fitness of a population through allowing beneficial alleles to find each other through recombination, here specifically through independent assortment of chromosomes. Okay, so at this point you should be able to explain how sexual reproduction creates genetic diversity and drives adaptive evolution, and why siblings are always different from each other even though they have the same parents. If you don't feel like you can, as always, feel free to watch part or all of these two videos again. Now, in the next few videos, we'll take a look at how abnormal numbers of chromosomes and chromosomal rearrangements can lead to human genetic disorders and how this stems from the events of meiosis. See you all then.